Good evening, everyone, and a warm welcome to the week 5 of the BioTalk series hosted by the Department of Biotechnology from St. Joseph's College of Engineering. Before we start, I request you all to follow a few instructions to ensure the smooth sailing of the event. Please do not introduce yourselves in the chat box. Kindly post any questions you have in the chat box and they will be answered during the Q&A session in the end. If you face any technical difficulties at any point, relate them in the chat box and we will look into it. We will now start with the auspicious note with the Tamil Thai Valthu. I now call upon the organizer, Dr. Chamundeshwari, to deliver the welcome address.
Good evening to one and all present here. A happy evening to all the faculty members, research scholars, industrialists, and my dear students who has actively registered and participating in this online webinar Biotech Talk series from various institutions across the country. This type of Biotech series was conducted successfully by our Department of Biotechnology for the past 1 month, and more participants have actively participated and got fruitful ideas related to the current cutting-edge research. As a continuation of it, today a Biotalk series of 5 is being conducted. So, on behalf of the Department of Biotechnology from St. Joseph College of Engineering, I welcome you all to the Biotalk series week 5 on the topic Microfluidics, Bacterial Intelligence and Biocomputers. I take immense and immune pleasure in welcoming our eminent speaker for today's session, Dr. AYYAPPASAMY SUDALAIYADUM PERUMAL, who is a postdoctoral researcher and course instructor in Department of Bioengineering from MC Gill University in Montreal, Canada. I also welcome the head of the department, my fellow colleagues, and my beloved students for today's event. Now I request head of the department, Dr. G. Srikumar, to address the gathering. Sir. I welcome you, sir, Dr. G. Srikumar, the head of the department, to address the gathering.
Good evening, my dear students and participants. I'm happy to see you all again in the Biotech lecture series, week 5. I should proudly say that our department keeps our students and other regular participants in line with the current trends in biotech research, even in the lockdown period. In fact, our aim behind this Biotalk lecture series is that pandemic outbreak should not be a hindrance for developing our knowledge and enhancing the intellect. So far, 4 lectures have been presented under this title, which were appreciated by all with positive feedback. For the sake of new participants, I wish to highlight some of the credits of our college and the department. St. Joseph College of Engineering, one of the top colleges in India, with the 160th position as per current NIRF ranking. It has got 26 years of high reputation in educational society with the NAC A+ grade accreditation and having 10 UG programs and 7 PG programs with an affiliation from Anna University, Chennai. Our Department of Biotechnology is more than 18 years old, having BTech and MTech biotechnology programs. Since inception, we have bagged 139 university ranks with 10 golds and 7 silver medals and achieve 100% pass percentage every year. The department has got sophisticated laboratory facility and fetched research center recognition from Anna University to do PhD and having more than 1.1 crores of rupees as government funding to conduct research. All along, through our lectures, we have been understanding the major outcomes of biotech research and its products and benefits to the society, etc. But today, the whole focus of the talk is going to be in a different angle. That is, micro level understanding of bacterial intelligence and its behavior, such as bacterial space sharing, partitioning, and motility characteristics, etc. How to use this understanding in design of efficient biological computing tool. How to use this revolution in the field of biomedical equipment fabrication. So this is going to be the angle of talk today. Behavioral biotechnology research explores the cognitive process within the microorganism and also the behavioral interaction between the organisms. Now, you could have understood that today's biotech lecture totally going to be out of box thinking in biotech research. To make us more informed in this unique field, today we have an eminent personality with us who is an expert in bacterial intelligence and its behavioral research, Dr. AYYAPPASAMY SUDALAIYADUM PERUMAL, postdoctoral research scholar from Department of Bioengineering, McGill University, Canada. Thank you, sir, for being as the resource person for this wonderful biotech series, week 5 session. At this moment, I wish to comment about our overwhelming response shown by the participants also. There are 612 registered participants that include 322 UG and 81 PG students and 173 faculty members. Also, the list includes 36 research scholars and industry personnel also. They are from 26 different states of India and 7 countries such as China, UK, Saudi Arabia, Iraq, Jordan, Ethiopia, and Zambia, etc. With this proud note, on the behalf of the patrons of the event, Dr. B. Babu Manoharan, Chairman, Ms. Jessie Priya, Managing Director, Mr. Sashi Shekhar, Director, Dr. V. Seshagiri Rao, Principal, Dr. Parvadavardhini, Dean, and on my own behalf, I wish to inaugurate this 5th BioTalk lecture series on bacterial intelligence and biocomputers. I also extend my special wish to the organizer, Dr. M. Chamundishwari, and my dear student coordinators from final years. All the best. Thank you. Over to Ms. Vatika.
The guest speaker for this session is Dr. AYYAPPASAMY SUDALAIYADUM PERUMAL. Dr. AYYAPPASAMY completed his bachelor's in biotechnology from Aruna Engineering College and did his master's in molecular pathogenesis and biochemistry from Center of Biotechnology in Anna University. He then obtained his doctorate in biochemistry and health from Université de Montpellier in France. He then became a research assistant at Johns Hopkins Medical University in Baltimore, USA. He has published papers in several well-renowned journals like Springer, Lab on a Chip, and Interface Focus. He is the recipient of various awards and was the semi-finalist in Dell's Social Innovation Competition organized by Dell Incorporated and University of Texas in Austin, USA. He was also selected as the Best Student of the Year in 2008 by Indian Society for Technical Education for the overall performance as a student and as a leader. He has participated in many conferences, symposiums, and seminars, both at national and international level. His expertise include microbial handling, protein expression and purification, soft lithography and microfluidic techniques, nanoparticle synthesis, and electrochemistry. Dr. AYYAPPASAMY is currently a postdoctoral research fellow at the Department of Bioengineering from McGill University in Montreal, Canada. Sir, it is an immense pleasure to have you with us today. I request you to take over the session.
Okay. Shall I go about presenting my slides?
Yes, sir.
Okay. Okay, so do you guys see my screen here?
Yes, sir, we can see them.
Okay, cool. Okay, cool. Thank you very much for inviting me to present this lecture or talk on some of the research that I've been doing in the past 3 to 4 years' time. At this point, I'd like to thank the management at St. Joseph's Engineering College for giving me this opportunity to discuss my research and the great amount of work that you put in to engage your students during this time of COVID-19 lockdown and all the extraordinary situation that the humanity is facing. Just I would like to have a confirmation from you, like if you are hearing me properly and if you're seeing the movies that's running on my slide.
Yes, sir.
Okay, cool. So also I'd like to extend my thanks to the Department of Biotechnology and Dr. Chamundeshwari for inviting me to this again. And let's go on with today's talk. So today I'd be talking about microfluidics, bacterial intelligence, and bacterial computers. So right now I'm working or I'm associated with Professor Dan V. Nicolaou at the Biological Microfluidics Lab. That's at the Department of Bioengineering at McGill University in Canada. So as much as you can see, the topic looks a bit clustered. So you have a field or you have an area of research that's microfluidics. You have another area of research that's bacterial intelligence. And then I come up with third topic that is bacterial computers. So much as it looks kind of clustered, you would understand why it is clustered, and also you would understand it is not clustered towards the end of the talk. So if you walk with me through the 40 slides that I have over here to describe the 3 topics that I have put as my title, you would be able to understand what kind of interdisciplinary research that generally bioengineering or area of bioengineering does. So that's a brief introduction about the topic, and let's quickly get on to the next one, which is where I live. So I was living in Chennai for the past 8-9 years. Right now I live at Montreal. So Montreal is a French English-speaking city, or it's a province that's in Canada. Which has many good universities around. And so those are some of the pictures that you see of the city, and as well as the distance or the time that takes to travel between Chennai to Montreal, which is roughly around a day. Okay, so the university where I do my research, so that's the university, the top figure that you see, that's how typically the university looks during the winter. It's a bit harsh city in terms of winter, but quite lively too. And that one that you see over down is the picture of the city during autumn. It's quite colorful during the autumn or during the fall. So McGill University, it's one of the high-end university that you have at in Canada, where we do really like some innovative, good pieces of research. And so the talk that I'm going to give today is one such. Let's quickly get on to the outline for my today's talk. So I would like to talk a bit about the introduction to bioengineering and interdisciplinary research, because just like it's difficult to define what is biotechnology, the word bioengineering is a bit difficult to define too. So I'm trying to my best to give my definition or my understanding of bioengineering and what is interdisciplinary research. So that's going to be one slide. And then part A of my talk is going to be about microfluidics. So probably if you haven't heard what is microfluidics, or if you have got a little bit of exposure about microfluidics, you would be appreciating those slides that's related to microfluidics. So people, since you guys, most of you are from the biotechnology, doing BTech or masters in biotechnology, you would have come across this subject that is called as fluid dynamics and mass transfers. during your second or third year. So this area, that is microfluidics, is an allied field of fluid dynamics. So you will know how and why in a bit of time. Part B of my talk is going to be about understanding bacterial motility, and as well as how bacterial intelligence could be deciphered, what makes bacteria more intelligent, or maybe more dumb in a way. So that's going to be the part B of the lecture, and part C is going to be the most voluminous part of my lecture. That's about bacterial computers. So the idea is, can we make a computer entirely biological? So that's the whole topic is going to be on part C. So I'm going to show you some real examples which I have been doing in the recent years, like the past 2 to 3 years, that talks about how a bacteria could solve a maze, or how we could make bacteria perform additions, subtractions, or even to a level of one of the highly complicated problem that researchers at mathematicians and researchers face, that is about how to solve problems that may not be able to be solved by an electronic computer, like a computer that you use in your homes, in your colleges, in your universities, even to the level of supercomputers. So how biological agents, particularly bacteria, could solve problems that are difficult to be solved by one of these. And the last part of my presentation is going to be some take-home messages and acknowledgements, and one of the venture that I have been doing in the time of lockdown. So I'm going to have a few words about it as well. So that's going to be the outline for today's presentation or today's talk. So let's move on to the introduction to bioengineering and interdisciplinary research. So one of the haunting questions that anyone would get as a biotechnologist is like, what is biotechnology, right? So it is quite hard to define biotechnology, and likewise, even bioengineering is kind of hard to define. The reason is you cannot name one single field or single area of science that you can associate with, because it is not. So if I have to define what is bioengineering, then I would say it is a science of everything. The reason why I have to say it's bioengineering or even biotechnology to some extent is science of everything is because any sort of research or any sort of understanding or learning that you want to take up in the field of bioengineering, it would encompass at least 2 different fields of science. Doesn't matter physics, mathematics, biology, or any new areas of research like nanotechnology, molecular engineering, molecular biomechanics, medical imaging, post-processing of imaging, artificial intelligence. Everything could be incorporated into a hybrid type of field, which is bioengineering. So what do bioengineers then work for? Most of the bioengineers, their motto as a bioengineer is to build or develop a tool, products, technology, or applications that will help them advance our way of living and well-being. It doesn't matter. It could be a diagnostic tool, or it could be a wearable device that you wear on your hands, which would interact with your system and transfer information from your body to an electronically readable device like smartphones, etc., or to complex biomedical equipments that could be used for diagnosing infections or diagnosing cancer, tumor, etc. So as a result, since the field is quite complicated with too many different areas of science coming together to have a happy marriage, so generally the area of researchers in bioengineering is quite interdisciplinary, meaning it involves more than one discipline of science coming together to find an application that would benefit human race or for the fellow other living creatures on the planet of Earth. So as a bioengineer, that have kind of excited me is when I go to my department to work, I would not be just interacting with biologists or molecular biologists, let's say, to be more precise, but also with mechanical engineers, electrical engineers, electronic engineers, computer scientists, people who are expertise in latest techniques in biotechnology, physics, biochemistry, and so on and so forth. So the area particularly the interdisciplinary research that I'm going to talk today in terms of biocomputers and bacterial intelligence is quite a melange of many different science areas. So that is sort of an introduction to what is bioengineering and what is interdisciplinary research. So why I call it interdisciplinary research, you will get to know more about it as we go through the slides one after the other. So that's the introduction. So let's go on to the actual research that I'm performing at McGill. So part A of my lecture or part A of my talk is going to be microfluidics. So as a fluid dynamics student or as a mass transfer and engineering student who have taken 1 or 2 courses on that, you would already have acquainted with the word fluidics. So fluidics is nothing but something that's moving and measuring something out of it. It could be the rate of flow, or it could be rate of transfer of materials, it could be rate of transfer of heat, mass, etc. So in terms of microfluidics, you do the same fluidic component that you would do in a large scale, but you would shrink everything towards micro level. So microfluidics is a process of manipulating small volumes of liquid. And the small volumes here I'm meaning is technically anywhere between few hundred microliters to as low as nanoliter or even less of femtoliters, which is 10 to the power minus 15 of a liter. So in order to manipulate such small volumes of fluid, you need to have devices that are micro in size or nano in size. And we are going to look into a few of those devices in the presentation as follows. So the next question is, do we have microfluidics operating in the world or in the earth, in natural systems? There are so many examples that I could give you where microfluidics, that's my alarm, sorry. So there are so many different areas, or there are so many examples I could give you where microfluidics is operating very efficiently in the world. One very classical example is the alveoli in our lungs, which is very essential for the transfer of oxygen that we inhale, or transfer of oxygen from air that we inhale to the other parts of the body. And so these alveoli, they have really very finite micro to nano sized capillaries where the oxygen transfer takes place. That's one very classical example of microfluidics in nature. There are several other such examples, like, for example, blood vessels in the body. So the blood vessels and capillaries that carry materials on and off inside the body from one end to the other, those are technically microns in size. And if you look into capillaries, they are down to few or less than 200 or 300 nanometers in size. And at these levels, when fluid is moving, what you call is microfluidics or nanofluidics. But most of my talk is going to be about microfluidics. Another example from the plant kingdom is the capillary forces that's acting in the plants, which is very essential for water absorption or water absorption from the soil. So you can imagine a tree as tall as, like, let's say, several meters, and water is going from the bottom of the earth towards the tip of the plant, which is all because of the capillary forces that's acting in these channels, which are generally micron sized. If you look into the plant anatomy, most of the material transfer happens through such micro systems. So here, there is this field, microfluidics, which is operating very efficiently in nature, doing wonders. And the human scientists or the humanity wanted to use these excellent materials that are operating in nature to be of use to mankind. And there came the field of microfluidics. So the whole concept of microfluidics is converting everything that's operating at a micro scale in nature to something that would benefit the humans. And as a result of working with materials in micro scale or operating fluids at micro scale, you would be able to decrease the reagent consumption. For example, if you have to perform, let's say, an ELISA reaction in a 96 well plate, you need to waste at least 200 to 300 microliters of the reagents. But with microfluidics, you can finish the reaction in down to less than 10 to 20 microliters, or sometimes even less. So as a result, the economic footprint that microfluidics would leave would be very less, meaning where you need 100 square meter of or 100 square foot of a table to perform your experiments, you can perform it on some device that would fit perfectly in your palm. So that's one of the benefit of using microfluidics. And because everything is going to the micro scale, the rate of heat transfer, the rate of catalysis, the rate of chemical reaction, everything is going to be accelerated. So instead of waiting for a reaction to complete for 1 hour or 2 hours, you could get your results well in minutes. So that's wonderful when it comes to diagnostics, for instance. Then there is a very high diffusion. You could study things very quickly, and at the same time that you can automate and integrate these microfluidic devices very easily into many operating devices. And also in terms of point of care devices, particularly devices that's used for diagnostics, you use a huge deal of microfluidics there. One very quick example that I could give you is the pregnancy testing kits, which is called as the lateral flow assay kits, is technically a microfluidic device. You have the strip, or you have the pencil-like material that is generally the pregnancy testing kit, where you put this in a scoop of in the urine that you have collected, and everything moves up on through this device, and as a result of it, you would be able to know if it is a positive or negative for pregnancy. So that's one of a very classical example of a microfluidic device operating when it comes to applications that's more related to microfluidics, and that's one of the applications that's point of care devices. You could deploy these devices wherever it is needed. Like, doesn't matter right from one end of India, one end of the world, like in Arctic regions, to African regions, you could deploy these devices very easily, and you could have the same experimental output or result that you would expect in a lab. So as a result of it, the whole field of microfluidics is moving towards how you could perform diagnostics in home, or how to contain the movement of people, or how to prevent, or how to have people tested themselves in homes, like home care or home point of care devices, or even better, to have implantable microfluidic or nanofluidic devices that would continuously interact with your body, with the bloodstream in your body, and transfer the information from the body to an electronically readable device. So that's where the field of microfluidics is going towards. And when it comes to microfluidic device, what scale are we talking about? How big or how small it is? What volume of fluids it could handle? That is represented in the chart that you see over here. So I hope you see my cursor. So that's basically the sizes that you could handle with a microfluidic device. So for a micro-sized device, which can be ranging anywhere between 1 micron to up to 1 millimeter, or even few hundreds of millimeter, you could handle volumes as small as from 1 femto or 1 picoliters to up to 1 liter or even thousands of liters. So that's the fluid handling capacity of these microfluidic devices, and these are quite sturdy, these are quite robust. You can use them on and on and on for a very long time. And anything that's less than 1 micrometer, technically 1000 nanometers, comes under the category of nano devices, which is separately another field, that is nanotechnology, which is not going to be the focus for today's talk. So the general understanding that I would like to give you at this moment is microfluidics is operating fluids in microscale, and the whole motto of microfluidics is to miniaturize everything and to probably transfer a lab which does a lot of analysis for diagnostics, for estimating different materials in the blood, like for example glucose, for other blood cells in the body, etc., into a device that you could use well easily at any point, particularly in homes and in those places where there are very less resources that's available. That's the motto of whole of microfluidics. So one other classical example I could give you is glucose monitoring tools that we have today in the markets. So for example, like 5-10 years before, if your father or mother or your grandparents had to check blood glucose, they would go to a clinic and they would ask, or the lab, and they would ask them to come before eating, after eating. Then they would take several days to get you a result of how much sugar you have in your body. That's quite complicated, and it's quite lengthy process, and there is a lot of possibilities for human errors. So the whole field of microfluidics now has transformed this glucose measuring analysis into something that you could carry in your pocket wherever you want. Just prick on your finger and you get the result of how much of glucose that you have in your body, or that you have in your blood. And as a result, you could well efficiently manage what you eat, what you should avoid at that time, etc., etc. So this is kind of the goal for one of the main goal for microfluidics, which is miniaturization, and that's going to be the future. And now we have devices, particularly several sensors or biosensors that can be implanted in the body, and you could read the device on your mobile phones. That's the impact of microfluidics, if I have to say. So how does microfluidics perform all this? What's driving all these materials in terms of microfluidics, or what is the physics inside microfluidics? That's going to be the next slide. So you have two types of flows generally. One is a laminar flow, which is quite uniform. Particles move smoothly, happily in one continuous fashion. And then you have another area, which is turbulent flow, where the particles move in a very irregular fashion. There is a higher Brownian motion. And at the same time, you cannot predict something out of turbulent flow effectively. When it comes to microfluidics, the fluids generally operate in a laminar flow fashion, meaning you have a very smooth sailing of particles and materials in a microfluidic device, be it a square channel or a circular channel, be it a specific type of geometries that you use in a microfluidic device. And as a result of it, if you want to understand how fluidics flow through a microfluidic channel, you have the figures at the left and the right. So the left one is actually showing particles or fluid moving in microfluidic device in a laminar fashion, and on the right you have the one that's moving in a turbulent fashion. Okay, so is that important for biological applications when it comes to microfluidics? Yes, it is very important. Most of the diagnostic devices, most of the biological testings that you want to carry out, or biologic-based interfacing devices that you want to perform in microfluidics, functions using the laminar flow, not the turbulent flow, which is other ways. So this is one very key property that's driving most of the microfluidics. And there are several other properties that are being used in several different applications, like, for example, electrical properties, thermal properties, pressure-driven properties, which has really enriched the microfluidic devices to a great extent, or the field of microfluidics to a great extent that it could benefit at almost every part of life. And if I had to impress you, I would say that in a lifetime of a person, at least once, or at least several times, if not once, they would have used the microfluidic based devices, and that's kind of the impact that microfluidics has created in a very short span of time since the first lecture that was given by Richard Feynman about there is a lot of space at the bottom, or there is a lot of room at the bottom, which he is technically meaning humans have to go towards micro and nano level of engineering. And microfluidics is one such engineering, which is basically trying to engineer at micro and nano levels. So next question is, can we fabricate this device, or how complicated is it to fabricate such devices? So in my opinion, I have quite good exposure to soft lithography, and as well as a fair amount of exposure to lithography. So based on my experiences, I would say microfluidics is something that you can very easily carry out in a normal lab in the present scenario, or sometimes even in homes if you want. So what are the steps that are involved in microfluidic based design and fabrication of a device? The first thing is you need to design the device, for which you need to have computer aided vector based designing tools, knowledge of that, for example, AutoCAD or CorelDRAW, etc. So you have these tools, you know a fair amount of idea about how to operate these tools, then you could fabricate, or you could make designs of the device that you want. And the devices can be as simple as just a straight channel, which probably you would see in the following slides, or I think you would see in the following slides, or it could be as complicated as several channels running interconnected with one another, running through one point to the other. So that's the first part, that is the designing. Then you have the lithography and soft lithography. So in the next step of lithography, you have to transfer the designs that you have made in the computer onto something physical. And here the physical material that's mostly used in microfluidics for preparing masters or wafers is silicon or monolithic silica. So this is the silica that you find in the soil, but it's a monolithic pure form which has been made into one single sheet of 100 microns to 500 or 600 microns in thickness. And on this top of this device, on the top of this substrate, you start making the pattern that you have made as a design in the computer onto it physically. That's the first next step. And after to that, there are several ways you could do that, which we will see in a very short, brief way in the next slide. And next to that, you have the soft lithography, where you make several copies of the design that you made in the computer, transfer it physically in the wafer. Whatever this design that you have, you make several copies of it, and that's soft lithography. And the last part is you peel the devices and you start fixing them on a flat substrate like a glass or something, and you can manipulate it to puncture it. You can fix tubings, etc., and then you can pump liquids through it. You can make chemical reactions, you can make biological reactions, diagnostic kits, etc., through the device. So this is a whole flow chart of items that you would take into account when it comes to fabricating the device. It sounds quite a lot of steps, but it is quite doable depending on the resolution that you would like to achieve. That's the whole point. And in today's next two slides, I'm going to talk specifically about lithography, that is how you make the masters, and the soft lithography, or how you make several copies of the masters, or how you make replica molding. So these two steps is very important to understand the remaining of my talk, and also a few more additional steps that would come in eventually. Okay, so when it comes to microfluidic device fabrication, you could close your eyes and just separate it into two categories. One is master fabrication, the other one is device fabrication. In the master fabrication, you make the master, which you will use on and on to make the replica. And the replica is what you call as the device fabrication, where you have the device ready for carrying out a specific function. So you could access to these articles or to the source of this information through the links that I've given you at each and every slide, wherever it is necessary. So this would help you get an in-depth understanding of what is microfluidics, if it is going to be something that you are interested to carry out after you read for the future. Okay, so now comes the first step, that is how you fabricate a master. So when it comes to master fabrication, you have two major domains of techniques. One is mask-based master fabrication, where you use a mask which can allow light to pass through at specific regions, which is basically the design that you want. It is actually shown in the extreme left of the slide here. And then you have the maskless fabrication, where you use high energy radiations, like for example ion beam, electron beam, laser light, X-ray, etc., to directly start writing patterns on silica material or plastics or on glass, etc. So that's the maskless fabrication. So in the mask-based fabrication, you draw the design that you want to make at micro scale in a computer, and then you print it on a OHP sheet. Okay, and then you would see something like what you see here with black background and white lines or white regions. So then what you do is you use a polymer that has been spin coated onto the substrate. So here the green thing that you see is a substrate on which the polymer that you want that is quite reactive to UV light is spin coated. So you use a spin coater where you add a drop of the polymer and you spin it at a high speed, so the fluid spreads out evenly. So on this spin coated device, you would place the mask, and then you would start the UV light for specific seconds, And wherever the UV light is falling on the mask material or the photopolymer, those regions would be cross-linked. Everything otherwise would not be cross-linked, so you can easily wash it off. And end of the process, you will have a device like this where you have the substrate, and then you have the green region, which is the photopolymer that has been polymerized. Now, when you pour something on top of it, you would be getting an exact replica of these channels onto the device otherwise. That's mask-based fabrication technique, and generally you could reach to a resolution of up to 8 micrometers, or sometimes even a bit less, like 6 or 7. It's higher actually, 6 or 7 microns, using the mask-based photolithography. So it's basically shown in this slide. And here you will be using UV light, sometimes visible lights as well. Then in terms of the other maskless lithography, you will be using direct electron beams, or you will be using ion beams or X-rays. Since these are high energy radiation, when you shine it to a level of micron precision onto a material, let's say for example a plastic or a carbon or even silica, it would at molecular scale disarrange or delocate the molecules in these regions. And because these are delocated or disoriented, you can easily etch them by using high corrosive chemical treatments. So at the end of the corrosive chemical treatment, you will again end up with something like this, where you will have channels that are not affected. Channels that are not affected anyway by the laser, by the high energy radiation that you use, and those areas where it is affected. So that's one thing. Later to that, you will be having the next step, which is the soft lithography. So now you have a master. What you do is you mix a polymer, and in this case, one of the most commonly used polymer is polydimethyl siloxane. So you pour the polymer on top of it and you let it cure. So you can let it at room temperature, or you could use 100 degrees, etc. to cure it. And after it cures, since the surface of the substrate that you use, which is generally silica, is actually hydrophobic. As a result, the polymerized material or the cured material would not stick to it. So which means you can easily peel it off. So once you peel it off, you have a device like that, and you can put holes on it, and you can fix tubings there, and you can fix this device onto the top of the glass cover slip. And in the end of that, what you have is a working device which is ready to be used for your specific application. That's the whole process of how you do the soft lithography. So that is what is shown in the right side of the figure here. So not just PDMS, you have several other polymers that you could use for this specific purpose, like for example, polystyrene, polytetrafluoroethylene, or polymethyl methacrylate. Depending on the polymer you use, they may need different ways to treat for curing. Generally, heat is used as the best curing, and in terms of PMMA, you don't need to even use the heat. You leave it at room temperature and it gets polymerized, or it gets cured. So this type of microfluidic devices are used extensively for multiple molecular applications. The diagnostic kits, HTS meaning high throughput systems. For example, the DNA microarray, several genome sequencing, the latest of gene sequencing techniques like nanopore sequencing, all of that use microfluidic based approaches. It is used in biodefense as well as for characterizing pathogens that's present in the atmosphere, in the food, etc. You could use it in molecular biology for flow cytometry, cell sorting, single cell studies, droplet studies, modern sequencing techniques. All of that uses microfluidic based devices. Also, you use that in microelectronics as implantable chips and sensors and wearable devices. You could perform almost any molecular biology based reaction on a microfluidic chip without any haze. So that's the impact of microfluidics. Now the big question, why am I talking about microfluidics when I want to talk to you about bacterial intelligence and biocomputers? The reason is, most of the process that I use for understanding how bacteria behave and what are the intelligence aspect that's associated with bacteria, I would be using a similar protocol. What I'd be doing is I would be having my masters that has been fabricated by one of our collaborator. Then I would be doing the soft lithography on it, where I will be preparing a PDMS mixture, pouring it on it. And at the end of the two important steps that I just described in brief in the previous slides, I would have devices at micro resolution, or to some extent even hundreds of nanometer resolution, which is actually representing some fine geometrical patterns. And in these geometrical patterns, I would let my bacteria that I'm interested to study through it, and I'll be looking into it, how the bacteria moves in it, what type of movement they have, do they try to go along the edges of the device or in the center of the device, do they get stuck a lot, do they go around in circles, so many interesting aspects I would be, and then what's their velocity, how much time they stay in these complicated geometries, etc., would be studied by a device that I would fabricate by microfluidics. And on top of that, I would be putting this fabricated microfluidic device on a microscope. Generally, I use fluorescence microscope because I have genetically modified my bacteria to express fluorescent proteins. Sometimes these genetical modifications are at plasmid level, where I do transformation to get my bacterial cells express the genetic proteins, or sometimes it's chromosomal level, where I would be directly incorporating the fluorescent genes into the genome of the bacteria, or to perform specific mutations that I'm interested in. For example, I would make a bacteria which would move only in the right direction or only in the left direction, or something that would not have a smooth and would not have any tumble mechanism at all, etc. So I would be putting the device bacteria around the device, and this is how the fabricated device would look like. And the orange solution that you see here is the bacterial culture, and this device is perfectly sealed onto the glass surface except those regions where the bacteria could enter, or these are called as the inlet points. And through that, the bacteria would be exploring these different types of structure. I would be recording everything that's happening with a microscope. Then I would be post-processing my images. That is another field of biology that I work with when it comes to post-processing my images. And this is generally image engineer, image scientist, or computer scientist that I work with during that point. And then finally, the output I would work with would look like this. This is very important because the output that I have, generally I could represent them in bar graphs or in numbers when it comes to velocity. But in order to understand the general moving pattern of the bacteria in the device, I would use something called as a density map, or in other ways, it's called as a heat maps. So what heat maps or density maps mean is, let's imagine you have your Google, you have your smartphone, and you look into the Google Maps. The Google Map would tell you how the traffic is moving in the road that you are going now, or if you want to check the traffic, you could do it on a Google Map. Now, it would show you in terms of colors based on the traffic. Sometimes in red, meaning it's more traffic. If it's blue, it's less traffic, so on and so forth. In a similar way, I would be studying the traffic of the bacteria in specific regions in the microfluidic devices using this specific approach that I'm going to show you now. So for that, I use a lot of density maps, and density maps are something very similar to how you see the pattern of movement of clouds in a satellite image when there is a weather report. So that's one of a simple way to get you to understanding of density maps. So most of my studies today that I'm going to discuss is going to be revolving around classical bacteria like E. coli, and one new organism which I would introduce to you today, which has the fastest growing rate in the whole of the earth, I think. And let's go on to that. So after I fabricate these devices, after I have my bacteria, I put them in the microscope, I run them in an experiment and see what happens. And based on the way the bacteria is moving, etc., I would be understanding what intelligence they have and if they have at all any intelligence. Okay, so that brings me to the part B of the lecture, which is bacterial motility and bacterial intelligence. So this is going to be one of the part of the work which is in kind of a communication into journals. So I will try to show you the information that's more representative to understand what is happening there. Okay, when it comes to bacteria, in your microbiology and molecular biology lectures, you would have learned about how bacteria move, what makes them moving, the electron transport chain, etc., etc. So in order to understand, that's enough for following this lecture. Bacteria has several different ring proteins in their plasma membrane, and these ring proteins are continuously operated by ATP, that is the energy currency of the cell, the adenosine triphosphate. And as long as ATP is being hydrolyzed by the ring proteins, the ring proteins are going to rotate either clockwise or anti-clockwise. Depending on the rotation of these ring proteins, the flagella that's attached to it is also going to rotate either clockwise or anti-clockwise. Why clockwise and why anti-clockwise? Because generally with E. coli, when the bacteria is attracted towards a nutrient or when it is moving towards something that it likes, then the flagella rotates anti-clockwise, and as a result, the bacteria would be thrusted forward. While when it comes to clockwise rotation, where the bacteria wants to change the direction or it doesn't like the nutrients that it is sensing, let's say there is an antibiotic there or there is a repellent in the direction that's going and it wants to change the direction, then these flagella, they would start rotating in the clockwise direction. And as a result of it, the bacteria would spread its flagella and would start to slowly change the direction. They start to tumble. So this run and tumble mechanism makes the bacteria move in the forward direction or change the direction. As simple as that. Most of my experiments that I'm going to discuss with you would be employing bacterial flagella operated bacteria or bacteria that's run by flagellas. And in order to study the bacterial intelligence in a way that humans could understand, I have to study how they move, because generally this is a kind of puzzle that you want to solve, and you would be understanding if the puzzle is solved or not by the way the movements are made. So in a similar way, I would be trying to understand how bacteria moves in these small confined environments, and based on their motility, I'm trying to understand how intelligent they are or if they have any intelligence at all. Okay, so for this specific study, I have used several different types of bacteria, and different types of bacteria or different species of bacteria have different types of flagellar arrangement. For example, the one that you see over here, it's called Vibrio nitrigens. It's one of the newly discovered bacteria. It has been discovered since 1970s, but it is the talk of the town recently in the past 5-6 years because it has one of the shortest doubling time. When it comes to E. coli, it doubles every 20 minutes, while this bacteria divides every 9 minutes at room temperature. So if you look into this bacteria very carefully, you will be able to see it has one long flagella, and this long flagella that's over here is responsible for the locomotion of this bacteria. These are called as monopolar flagellated bacteria. On contrary, you have Escherichia coli or E. coli, one of the most commonly used laboratory bacteria, which has flagella spread all around the body. They don't have one single flagella, but multiple flagella. Also, you have another type of the same genus, Vibrio, which is Vibrio fischeri, which has multiple flagellas originating at a polar end. Likewise, Pseudomonas putida, one of the organisms that's used heavily in biotransformations and bioremediation, etc. And the bacteria that I show in the middle here is called as the Magnetococcus marinus. It's a very special bacteria because it can get attracted or repelled to the magnetic poles. And you can devise its movement, you can control its movement by using magnetic fields. And this guy, he has like around 9 flagellas distributed at 2 bipolar regions. And these are quite fast bacteria. They run at around 200 microns per second, which is at almost 200 times its body length, meaning the size of the bacteria is around 2 to 3 microns. And every second it can go 10 times its body length, 100 times its body length, while other bacteria can go anywhere between 10 to 15 times its body length. So I have used all of these bacteria for my understanding. And why these understandings are very essential is because most of the single genomic studies, droplet microfluidics that involve microbes, they have bacteria that's moving in the solution. So you need to understand how they move in these confined devices. Then it has a lot of implications in bio-inspired applications. Like for example, if I have to give an example, the honeybees, they're generally called as cicadas. They have a wing which is quite antimicrobial. The reason for antimicrobial property of cicada wings is because it has a lot of tiny pin-like structures protruding on the surface. And because this pin-like material that's protruding on the surface has very high surface tension, when a bacteria tries to sit on them, it pierces through the bacterial membrane and tears them apart. And as a result, the cytoplasmic contents would be released, leading to the bacterial cell death. humans are trying to mimic this on antibacterial surfaces. And in order for bacteria to colonize such rough surfaces, rough membranes, etc., we need to understand how they do that. And for that, we need to understand how bacteria move on these surfaces, how they can adapt to these surfaces. And for that, the motility related studies or how bacteria moves is important. Also, you have the next thing, which is microbial space partitioning and intelligence. That's going to be the crux of this part B of the lecture. And at the same time, you also have molecular pathogenesis and intercellular pathogen related studies. For example, when a bacteria is colonizing the human body, it's either living inside the cell or between the cells. And these spaces are generally micron sized. For example, inside the cell, they live in anywhere between 50 to 100 micron sized cells normally. And if it is between the cells, it is less than few microns. So as a result, how bacteria try to adapt these environments, how do they negotiate the space around them, how do they colonize these environments, that's going to be the other area why understanding bacterial motility is important. Because they say that researchers have found that if a bacteria is able to move, they have a higher chance of spreading and as well as higher chance of virulence associated genes, which could cause a stronger infection than compared to those bacteria that do not move that much. So in these aspects, trying to understand bacteria moving in the network in this in vivo condition is very essential. But having an in vivo condition replicated in the lab is very difficult. So you have the microfluidics, where you could take the geometry of the cells, geometry of the space in a cell, and try to reproduce it in a microfluidic device and leave bacteria there and see how it behaves there. So in these aspects, the topic that I'm going to discuss in part B is going to be relevant. Okay, so one of the first type of study that we did is with a very simple plaza-like unit, which is a square chamber, which has two inlet and 2 points where the bacteria could come in and go out. And when you leave the bacteria to explore this square region, we look into how the bacteria moves in this square region. So for example, in this slide, if you look into the top one, that's over here, the green and the black region, you can see that. Let's first focus on the first green region, which is basically a square like plaza that you see over here. And you can see a lot of lines that are moving around everywhere. And if you carefully notice here, you can see that the bacteria doesn't go to the boundary at all. It hits the boundary and then it jumps from there. It ripples from there. So the main reason for it is it kind of looks like how a ping pong ball works, right? Like you put it, it jumps everywhere. It doesn't go along. So go along the wall or the edges of the device. So this kind of ping pong type of motility is very characteristic to this type of bacteria called as Magnetococcus marinus, which is basically bacteria having two polar flagella, bundle of flagella. While you compare it to the other species, Vibrio nitrogens, you can see that they try to move along the edges of the device, that is along the boundaries of the device. And the reason they are moving along the boundaries of the device is because their motility pattern is more motivated towards moving along the edges. Okay, so this specific type of movement, we call it as wall dependent movement and wall escaping movement. For instance, the Magnetococcus marinus, since it doesn't go along the edges or the walls of the device, it's called wall escaping movement. And in terms of Vibrio nitrogen, it's depending on the wall for its movement, and so it's called as wall dependent movement. So depending on how the bacteria moves in the wall dependent and wall independent fashion, they have different applications. And this is one of the first type of intelligence that we noticed when it comes to how bacteria try to interact with their nearby surroundings or nearby walls. So the motility pattern that you see here has been superimposed over one another in the form of a heat pattern. And if you look between these two density maps or heat maps, you can see that in terms of the wall escaping bacteria, that is Magnetococcus marinus, they try to move around everywhere in the structure. You have a very high saturation almost everywhere. While in terms of Vibrio nitrogen, you see a lot of dark regions, which is corresponding to no motility at all there, or bacteria haven't visited those regions at all, compared to that of high density regions that you see over here, which is basically because the bacteria try to associate themselves along the wall for its motility. And particularly, you can notice that along the corners, the bacteria try to accumulate a lot. So This type of understanding where the bacteria tries to accumulate could be used for effectively trapping the bacteria. Like if you want to trap the bacteria that is in let's say in a body fluid like urine or serum or blood, you could devise a device, you could design a microfluidic device and let the fluid pass through it, and you can effectively look in these corners to see if there are any bacteria that is accumulated. So this could be a diagnostic application, but that's not going to be what we are going to do in what I'm doing in my research, but it's trying to more understand the bacterial hotspots, or where bacteria try to accumulate in excess, and how their motility is different along different areas of the channel. And this is again interesting to study because you can study to a really confined environments. For example, this device, it has a 100 micrometer by 100 micrometer x-axis and y-axis lens, and the height of the device is just 5 microns. So the bacteria is typically quite highly packed in this device, and as a result, you could try to closely monitor them, how they behave in such constricted environments. It's like you could imagine you put a human in a room and lock the doors, and you have a cam that monitoring what he is doing and how he is behaving, etc. So that's one of the interesting thing that we looked by just letting the bacteria in really a very smooth device. Then in order to give you an emphasis of how the corners could be used, because I told you here that Vibrio nitrogen, they try to get accumulated along the corners. If you look into this device, it has several corners in the form of arrow marks purposefully made in order to see if bacteria could be trapped in these corners that are in this device. As a control, we have here straight channels. So if you look into this movie, you can see that this green line representing bacteria moving in the straight channel, it's moving without any hassle. It's moving straight through it from this end to the other end. But if you look into this structure, which is having multiple of these arrows, multiple of these corners, bacteria can only move easily in this direction, that is from left to the right. And when it is trying to move from the right to the left, it gets easily trapped in the corners. So this is what you're going to see in the slide here. So you can see bacteria getting stuck at the corners is basically because you can see one right there, yellow line. It has got stuck there is basically because they like corners, and specifically bacteria like which follow the walls, they like the corners too much. And this can be one of the important strategy that we could use for trapping bacteria. And in fact, we do use this specific method to trap bacteria in a bacterial computers, which I would show you in the following slides. And another example is we were thinking, can we let bacteria move in only one direction? Can we control the movement of the bacteria in one direction only? So for that, you have a design that we made in a computer that's over here, and then you made it as a master. So this heart-shaped structures that you see over here, these are called as rectifiers. The reason they are called as rectifiers is because rectifiers try to let the movement of liquid or particles in one single direction. These type of rectifiers are also being used in the hearts. Let's say those valves which only allow the flow of blood through one direction, though those are controlled by some membranes. So what happens in these type of rectifier is a bacteria could enter freely into the heart-shaped channel and continue its motion in these straight channels, but they cannot go back, because if they try to go back, they would be again pushed back into the network in the forward direction, that is from top to down. This is what you are going to see as an example here. So you see that the bacteria is moving straight without any issue, and you can see right one there which tried to go back, but it got locked in these rectifiers. So as big or as large as rectifiers and as much as you can lower traffic you could have, you can very effectively let the bacteria to go in one single direction. Let's see the movie again. So you have the bacteria which tried to move in this direction but got stuck. It's struggling to go forward, but it could not. At some point, you can see that the bacteria would continue to change its direction and move forward. You can see that it moved forward and now it goes out. So that's one simple example of how we could control the movement of the bacteria. One important point here I have to say is the reason the bacteria looks kind of light-like or white particles is because they are expressing fluorescence. And it's a monochromatic camera that we use in the microscope, so you see them as black and white or in grayscale. Well, you can give them false colors like the one that you saw in the previous slide here. where the bacteria look reddish because they are expressing M-cherry protein, which is a red fluorescent protein. All right, so this is about how bacteria could be controlled in our geometries, and these are based on the understanding that we got through experimenting bacteria with very simple structures. Now, like that, we have used several different structures, geometries to understand how bacteria behaves. And we have gained a lot of information on that. And these are exactly being used in some of our biocomputation devices or in some of our bacterial computers, if I have to say. So, based on all of this, what I can give you as a summary at this point is we can use we can design microfluidic devices by using the field of microfluidics, which is an engineering and physics applied field, and then using biology like bacteria, you could let them in these microfluidic devices, in these constrained spaces, in these restricted spaces, and then see how they try to negotiate with their environment, how they negotiate with the surrounding spaces like the walls, the floor, the ceiling, etc. Now, why are they useful? Because the microfluidic device that we are going to use, or one of the steps for a biocomputer to work, or for making a biocomputer to function, we need to understand how bacteria behaves in these small environments. The better the understanding we have about bacteria in these micro confined environments, we have a better understanding of how our biocomputer or bacterial operated computer is going to look like and is going to behave like. So that's the main motto or reason for using these simple geometries, traps, etc. as a preliminary studies. The next important thing is the part C of the lecture, which is going to take some time. It's very interesting if you continue to follow through it. Okay, so this particular part C of the lecture talks about three things. One, about mathematics. Two, about how computers operate. And three, about how we could use bacteria to or other biological agents to have a biological computer operating. Okay, so that's going to be the 3 points that we are going to discuss in the part C of the lecture. So first, let's crack down what is the mathematics part. You would have done in your schools, like right from your first standard or second standard, addition, subtraction, multiplication, and during your higher secondary, you would have done square roots, squaring, cubing, some simple formulas to calculate volumes, areas, etc. All these type of mathematical calculations fall into the first category of problems called as a polynomial time problems, meaning within an estimated time, you could arrive at the solutions for the problem. That's polynomial time problem. These are quite easy problems. You can most of the time calculate them mentally by just thinking, or sometimes using a calculator, or sometimes using a laptop if it is absolutely necessary. And depending on the number of inputs you give, you could have a solution in exact number of replica of the time. If you have 10 inputs, if let's say if one input takes 1 minute, 10 inputs would take 10 minutes to solve. It's as simple as that. This is polynomial type of problem. There is a second and third degree of problems, which is called as the NP complete problems and exponential time problems, or NP hard problems. The NP here is the abbreviation NP stands for non-polynomial, meaning whatsoever you do, you use a supercomputer, you use a quantum computer, you use any type of computer, you will not be able to attain the solution in an estimated time. Or in a way, when the problem size increases, if you have one sample input, you may solve it very quickly. You have 10 sample inputs, you may solve it very quickly. You have 100 sample input, it would take quite some time. If you have 1000 sample input, then at this point, the computer, whatever the efficient computer that you're using, the computer would crash because the computer goes intractable, or you could not track the solutions to the problem for such large computation. And most of the time, for simple aspects, like simple example, like simple day-to-day life, we use a lot of NP complete problem, which I will explain you in the next slide. So the reason the computer cannot operate efficiently for solving NP complete problem is because computer operates sequentially, meaning when you give 10 numbers as an input to a computer and ask it to find all the possible additions for the 10 numbers, the computer can only solve one solution at a time. Meaning, if I give you three numbers, 1, 2, and 3, and I ask you what are the possible all additions that you could get out of it, you can say 1 plus 2, 1 plus 2 plus 3, 1 plus 3, 2 plus 3, so on and so forth. Now, in order to arrive at this solution, the computer would try to explore the solution one at a time, and it will go on and on. And imagine you have 1,000 different numbers there. In that time, at that point, a computer may not be able to explore all the solutions at all at any point, because it would lose its memory, it would lose its tracking nature, etc., etc. As a result of it, when it comes to NP-complete problems, computers, because of their sequential operation, it cannot solve such problems. The solution to that is parallel computing, meaning instead of one computing power trying to solve one problem, if you have 10 computing powers, or if you have 100 computing powers trying to explore all the possible solutions, then you will be able to arrive at the answer 10 times faster, or 100 times faster, or 1,000 times faster, depending on the number of computing units that's trying to solve. So stay on with me on this point. You will get to the understanding of it with the following examples. Let's say you are packing your bags for a trip or for your studies abroad, and you have a 23 kilogram that you have to pack for the flight, and now you are going to think in a combinatorial way by let's have this or let's have that, let's have this many number of jean pairs over here, this many number of t-shirts over here, etc., etc. So you would be mentally calculating how efficiently you could pack your 25 kilograms or 23 kilograms so that you would carry a lot of items. At the same time, you would also have a bag that's packed in a small way. So this is one very small example of how NP-complete problem works. You have so many possibilities, and you need to explore all the possibilities in order to say you have arrived at the optimum best possibility. And unless and until you do not have this optimum all the solutions explored, you cannot say you have solved this type of problems. So because of that, because the computer has to explore all these problems, it becomes really difficult for the computer to solve efficiently. Another example I could give you on packing is, imagine a thief is trying to pack his bags for his loot. He has golds, he has diamonds, he has rubies, he has silver, he has steel, he has copper. He has to imagine which would, if he would pack in his bag, he would get the maximum value that he could loot. So these type of combinatorial problems are very quickly worked out in our brains, because our brain sort of parallelly computes everything. Our brain, in our brain, our neurons compute much effectively in a parallel way. But computers do not, because computers operate sequentially. Because the computers operate sequentially, and because there are so many different solutions that you need to explore, and you need to explore all of the solutions for sure in order to say you have arrived at the best possible solution, because of the voluminous number of conditions that are involved, a computer cannot solve it at all. Another example I could give you is very easily associatable. It's called as, originally it was called as a Hamiltonian pathway problem, or sales problem, or a postman problem. This was one of a very critical problem that researchers tried to solve during the World Wars, because the postman who is carrying letters, important information, have to travel multiple locations in the same city in such a way he should not go to the same street again. He should not, you should use as little fuel as possible. He should use as less time as possible, and he should deliver hundreds of letters in a day. When all these conditions comes into existence, then there is a huge explosion of combinatorial mathematics that's involved, where you need to work out different combinations, A, B, C, D, etc. in different orders. And at that point, the whole part of computer calculations or computing would explode to voluminous levels that you will not be able to have a solution using computer. Even now, such Hamiltonian pathway problems, salesman problem, is a very complicated problem when it comes to mathematics, and people use supercomputers to solve it. The problem might sound very trivial, just additions, but when it comes to thousands of numbers together, the combination you would have to explore would explode so much that it's very difficult to solve these problems in a polynomial time or in a restricted time. You need to either run it until eternity, or your computer would crash meantime. Another example is when you're again booking a flight ticket. Let's imagine you want to make a trip around India, starting from south, go around the north and come back to the south, and you wanted to visit 3 cities. Yeah, 3 cities you can manage. Okay, I go here. Let's say I start from Chennai. I go to Goa. I go to Srinagar. Let's say I go then to Calcutta. Then I come back to Chennai. So it's a easy way. But imagine you need to make the trip for 20 different cities, and in such a way that you cannot travel one state again. You cannot pass one city again. You need to consider the shortest time you can spend on each city. You need to consider the travel expenses, the fuel expenses, the staying expenses, etc. The combinations are huge. So you cannot solve such problems. Our mind cannot solve those problems in any time, and despite being it's parallel. And likewise, there is no question a computer could solve it because they are operating sequentially. Another example I could give you is when it comes to allotting students in a hostel. Like, for example, you cannot where the biology comes into play. That is where the different biological agents comes into play, which you will see in the following few slides. So according to Richard Feynman, one of the great physicist, during his plenary lecture, he told that for Nobel Prize, he told that if there is a technology that could solve NP-complete problems, then that technology would be called as a demigod, if not God. Meaning the technology that could solve NP-complete problems would not be just sitting in one planet. It would explore everywhere around, different galaxies, different parts of the universe. That is the impact of NP-complete problems. These are as trivial problems, but very important problems that we don't have a solution for it. So now I'm going to show you how we could solve such problems using biocomputers. Before I go to bacteria used for solving such computers or such problems, computation problems, I'm going to talk to you about how we could use Physarum, which is a fungi, to solve mathematical problems. So here, what you see on the screen is different dots of sources of food, and the yellow spot that you see here is actually the fungi or the Physarum, which will try to grow in time. So the whole study was conducted for 26 hours, and the whole region that you see here is actually the map of Japan. And these are the important cities that you have when it comes to the map of Japan. And using Physarum, the whole network, railway network of Japan was designed, or was again confirmed that that is an efficient design to reach all different cities in best time. So this is one of the very classical example of how we could use biological agents to solve one important mathematical problem of trying to find the best possible routes across different cities. So this is the actual Tokyo railway network, sorry, the railway network in Tokyo. And this is a network that was sorted out by using a fungi called Physarum. This paper was published in a Nature journal back in 1990s. So the field of biocomputation is not new. It's there for more than 20 years now, and a lot of people are working on it to efficiently compete with silicon computing or use electronic computing. However, we could not use Physarum because Physarum tries to move only towards the nutrient sources. If there is no nutrient sources, they don't move. If you look here, they first try to move everywhere, but the network only strengthens on those spots where there are nutrients, all these yellow spots that you see here. As a result, because the network would be biased, you may not be able to explore all the possible solutions, but only specific solutions. As a result, fungi is not the best model that I would use for an NP-complete problem. But this is one of the first instances how we could use a biological agent for computing. The next one is coming with molecular biology techniques. It's called as DNA computing. So here, what you do is simple operations like ligation, restriction, PCR-based amplification, and then sequencing in order to solve mathematical problems. You might be wondering, I thought these techniques were used only in biology for disease diagnosis or trying to understand the biology of different organisms. But when it comes to DNA computing, you can use them, use these fragments of DNA for computing mathematics. How it works is you will have different fragments of DNA, let's say in different colors, and you call it as 1, 2, 3, 4, etc. And you would be basically performing ligation operations in a single test tube. And if you have thousands of molecules of DNA, all of these thousands of molecules of DNA is operating for ligation, or is getting ligated with each other in different orders. And as a result of it, every ligation you perform, it's an addition. Every restriction you perform is a subtraction, so on and so forth. So when you perform such process in real time in a test tube with thousands of molecules functioning at the same time, you achieve a maximum parallel computing. You achieve a maximum parallel computing point. And as a result of it, at the end of the whole process, you will look for a sequence of DNA which will have all the 4 fragments in a specific order that you may be looking at, or in an order that you would like to explore in the future. So based on this DNA type of sequence, DNA computing approaches, you could solve very easily problems that a computer cannot solve. However, in order to solve a problem that a computer that could overtake a computer, you need DNA molecules in a level that is equal to the mass of Earth. Let's say, for example, a computer, an electronic computer could solve problems of up to 20 variables very easily, and 30 variables it may crash. In this situation, when you use DNA sequences as a DNA computing, you might need DNA fragments equivalent to the whole mass of Earth. So this is not possible, and as a result of it, DNA computing did not take a big fly. It did not get through. The next type of computing uses molecular motor proteins, actin, myosins, cytoskeleton filaments, etc. This is one of the pioneering field where our group is working. And also, I'm going to talk to you about my experiences with bacteria in terms of making bacterial computers. So in terms of molecular motor proteins and bacterial-based computing, we are going to use a specific technique or specific technology, which is called as network type computing. So network type computing works very simple like this. You have a mathematical problem, and if you can have a mathematical problem represented in a graph-based method, which is called as graph theory-based coding of problems, if you can convert the mathematical problem into a graph, and if this graph can be converted to a physical microfluidic network, and if you have a biological agent that you could explore these physical networks easily, then what you have is nothing but a working operational bacterial or operational biological computer. So in order for network type computing to function, you need 3 things. One, you need a mathematical problem that can be encoded in the form of a graph, okay, like X and Y axis, X, Y, and Z axis. And then this graph should be translated into physical microfluidic networks, okay? And you will see examples in the following few slides. And then this physical microfluidic network that's representing a mathematical problem in the form of a graph should be explored by the bacteria. And if these 3 conditions satisfy, it's something like the Asimov's rule. If these 3 conditions satisfy, you will be able to have an operational biocomputer. And that's the whole point of this lecture. So one of the first approach that my boss or my professor over here, my mentor over here tried is he made a problem called a subset sum, which is a very simple addition-based problem. And he used molecular motor proteins to explore the network and find solutions to it. So that's one of the first example. And how it works is you have the bacteria. So sorry, you have the let's assume this is a microfluidic network, and this microfluidic network has several lines or channels, and these channels are interconnected with each other in a specific fashion or in a specific algorithmic way. And what you have is you have an inlet point where you let your motile biological agents to enter and explore the networks. And the moment when a bacteria or when another biological agent like actin, myosin, or cytoskeletal filaments like microtubules comes out of the exit here, you say that one solution has been explored. For example, if an agent is coming out of one of the line over here or channel over here to the exits, it means that solution is solved. Likewise for the remaining. Depending on how bacteria or other biological agents explore the network, you could solve the problems faster or slower. Okay, so this network that you see here is one similar example of a complex microfluidic network with several lines connected with each other. And there are some specific junctions that's operating specifically in this network. And white parts that you see moving over it, it's called as those are the molecular motor proteins. And these are the exits where you see the bacteria would end up. And each exit is coding for a corresponding solution. And everything that you see in green is actually a right solution. Everything that you see in magenta or pink is actually a wrong solution. Depending on which exit the bacteria comes out, you can easily say if it's a right solution or a wrong solution. Generally, if the bacteria follows the rules that you have incorporated in this microfluidic networks, which is basically the junctions, you would be able to understand if the bacteria is properly solving the problem or not. And also normally those right exits would be explored more than the wrong exits if the algorithm that you have incorporated into this network is being followed. So this is a concept figure of how a network computing would work, and this is how the network computing in reality would look like. And we will talk about it more in the next few slides. So let me start with giving you a very simple example of a bacteria solving a problem. Okay, so you would have in your childhood you would have solved a maze where there would be a bunny at one end and you would have a carrot at the other end, and you would be drawing with pencils, finding the route for the bunny to find the carrots. So this is very simple example to estimate or understand the cognitive power of any living creature in the world. You use these simple puzzles for exploring the cognitive skills of different animals like rats, mice, chimpanzees, elephants, ants, whatever the animal that you have. So we are going to see if this classical problem that you would have solved when you were kids is actually an NP-complete problem, because as the problem size increases, the time taken to solve it also increases. Let's say, for example, the first one that you see over here can be easily solved even without using a pencil. This one you need a pencil probably to solve it. This one it would take some time, probably several minutes. And this one, it's very difficult for you alone to solve it. Maybe if you have 2 or 3% solving on the same maze at the same time, you could solve it faster. Now, imagine you have to solve all these four types of mazes of higher complexity as you move from left to right by one person. Okay, what you would do is you would start for this maze, you would start at the entry point that's over here, and you would go until you get blocked. Again, you would go back, and then again you will find another route, and then again you will find another route until you find a way out here. This is a classical example of sequential computing. Okay, imagine you and 10 of your friends are trying to solve this maze at the same time. So 10 of your friends would explore 10 different routes at the same time, meaning one person operating a very big mask or trying to solve a very big maze versus 10 solving the same maze together. You obviously could calculate who would win faster or who would find the solution faster. That's going to be 10 members working on the same puzzle. So this is what we are trying to see here. That is, we are going to let the bacteria explore these mazes and see if they can solve the puzzle or not. So for this work, again, I use 5 different bacteria, namely Vibrio fischeri, Putida, Marinus, Escherichia coli, and Vibrio nitrogens. And my maze is very simple, but it's a bit different. What happens is a bacteria could enter either from this end or this end, and it has several blocks in the region, and there is no unique solution to this maze, meaning there is no one single solution, but infinite number of solutions. A bacteria could try to enter this way, and just take this yellow line or yellow lane to reach the exit, and this is one of the shortest exit that you can expect. Or the bacteria could take this red line or red path to go out of the network, and this is one of the longest path. But there are technically infinite number of solutions, and the bacteria could spend its eternity in this network if it has to. Now, what you see here is bacteria that's moving in the network, and you see here as a fluorescence signal. So what you see here in the red line is actually a bacteria entering, and it's finding its way out. So this is the way out, and this is the entry. You see other bacteria exploring different combinations, different networks at the same time too. Okay, so but the point I want to emphasize here is this red line that you see is one single point that the bacteria is trying to explore. Then you have another network that's over here, again explored by multiple bacteria. And here you see one bacteria trying to go on and on in the network, find new routes, and finally find a solution and go way out of it. But at the same time, one of the bacteria is exploring the right route. There are also several tens of bacteria that's exploring the route at the same time. This means that you have a maze, and you have to compare each bacteria to that of computing power, one computer, and each bacteria is equivalent to one computer, and every one of them is trying to solve the problem on and on and on. And imagine if you have a large network with thousands of possible solutions, and you have thousands of bacteria that's running and exploring the network, then what you have is one solution, one computer, meaning you amplify the rate at which you could find solutions. And this type of example where multiple bacteria try to find the solution at the same time is called as parallel computing. This type of parallel computing has been previously also achieved with molecular motor proteins, but it had some drawbacks, and that is why bacteria is coming into the play here. Okay, so here what you see is the same maze with entry and exit, and you have different heat maps of different bacteria, and you can see obviously that the heat maps don't look the same because different bacteria have different ways to explore the network. And generally we use the velocity, the time that the bacteria spends in the network, time taken to find the routes on an average, etc., to be the qualifying conditions to say if a bacteria is intelligent or not. For example, if you look into one specific case study here between Vibrio nitrigens and Vibrio fischeri, these two are belonging to the same genus, Vibrio, but are from different species. They have a varying velocity. That is, Vibrio nitrigen is 3 times slower than Vibrio fischeri, but the time taken to solve the network on an average is quite the same, meaning bacteria try to interact with the edges, interact with the network regions, interact with the confined spaces in different ways. Another example is between Vibrio nitrigens and Marinus. Vibrio nitrigens is one of the slowest bacteria that I used, and Magnetococcus marinus is one of the very fastest bacteria I used. So their velocity, if you compare the velocity, Vibrio nitrigens velocity is at least 12 times slower than that of Marinus. But if you look into how quick Vibrio nitrigen would solve, it is 2 times slower than that of Marinus, which means the velocity doesn't play a big role when it comes to negotiating with the environment, but it is mostly the the way the bacteria is trying to negotiate its space around it, trying to interact with the space around it. So the question is, why the slower microbe like Vibrio nitrigens or E. coli solve much faster compared to that of the faster microbes? The answer to that is, as I was telling you before, the way the bacteria interacts with its surrounding, interacts with the surrounding geometries. And this, we call it as the geometrical intelligence. And if you further try to look into how the bacteria tries to negotiate with the surrounding environments, surrounding geometries, then what you have is the algorithms that you could use for better traffic controlling, or for better planning, understanding better how molecules move inside our cell, etc., etc. So these are potentially the application areas of biocomputing. And one simple problem I showed you here is maze being solved by bacteria. Now let's step up our problem. Let's go to a bit complicated problem. I'm skipping that. Let's look into a bit complicated problem. So the complicated problem that I'm going to talk to you now is subset sum problem. It is a truly NP-complete problem. Computers may not be able to solve NP-complete problems that are larger than 30 or 40 sample numbers in size. So for this specific approach, we use 2 bacteria, that is E. coli and Vibrio nitrigens. The reason we use them are they have best laboratory conditions that we could manipulate them. And at the same time, they could be genetically engineered. They are self-propelled, meaning you don't need external stimulus to move them. They can move on their own, and you could genetically modify them extensively. And importantly, these bacteria could divide. So division is again another important criteria here, because imagine you let 1,000 bacteria into the network at one time, and imagine the network is sufficiently large, and let's say it takes 1 hour for a bacteria to run from point A to the exit in the network. And in this situation, bacteria could divide 2 or 3 times, meaning you start with 1,000 bacteria, and assuming all the bacteria divide, you end up with 6,000 bacteria in the end. Meaning you start with 1,000 competing units, and you end up with 6,000 competing units. Meaning, depending on the size of the problem you use, you could amplify the computing power, which means you could improve the parallel computing capacity, and as a result, you could arrive at the solutions much faster. In the starting of the slides, I was telling you the area that is mostly with bioengineering is interdisciplinary. And this is one classical example, because here 3 fields are involved: mathematics, engineering, and biology. So this is a simple way how to represent graph theory into physical networks. So you have a problem, you put it in a graph in x-axis and y-axis, you transfer them into a physical network, and this network has specific algorithmic junctions. or specific gates, if you want to call it, which will have to follow specific rules. And if the bacteria follows the rules in these gates, then they will eventually end up at specific exits. For example, this problem here is 1, 3, and 4, meaning what are the various possible combinations that you could get by adding the elements 1, 3, and 4. It could be 1 plus 3, which is 4, 1 plus 4, which is 5, 1 plus 3 again 4, 3 plus 4, which is 7, 1 plus 3 plus 4, which is 8, so on and so forth. So out of all the 8 possible solutions, 8 exits that we have, there are 1, 2, 3, 4, 5, 6 different solutions, and 2 different solutions, namely exit number 2 and exit number 6, are wrong solutions. Okay, so when you let a bacteria in this network, they would try to go out of the exits mostly 0, 1, 3, 5, 7, and 8. And if they follow the rules that we have employed in this network, they will not come out of the exits, namely 2 and 6. And this is a very classical example of how we could convert a graph theory based network problem to a physical microfluidic network. How this microfluidic network functions, we can understand it, or how this biocomputation network type biocomputation device functions, we can understand it by a simple quick animation. So we call it as SSP because it is subset sum problem. And you have a microfluidic network. This is the device, and the bacteria enters through this point, which is called as the inlet. It goes through several of these channels and it comes out of the exit, and each exit corresponds to a solution. And like I was telling you, these are the traps. It has square ends. So when a bacteria is entering the trap, it gets attracted to the squares, so they cannot pass. This network functions with 3 different types of junctions, or these are the algorithms that function in the network. First junction is called as a split junction. In this junction, when a bacteria is entering, it has equal chance to go either left or right. If it goes right, it adds 0. If it goes left, it adds 1. And any other direction, like you see here, are wrong directions. A bacteria can only go in the left or the right and no other way. The next type of junction is called as the pass junctions. So pass junctions, these are very simple junctions, which is plus type junctions or crossovers. And in these junctions, the bacteria has to continue in its path. It cannot turn left or right. If it is going from here, it has to continue in this direction only. Any path that, like U-turns, or going from left to right or right to left, is considered a wrong turn, and these are technically bacteria that do errors. And then you have the third type of junction called as the joint junctions. So these joint junctions, they are very specific junctions, which we will explain in a bit. So when a bacteria is trying to take this route, it is taking a left here, so it is adding +1. And then it's taking right, so it is adding 0, 0, 0, so it ends up in the exit 1. And so 1 is a element that you used in the combination that you used, a combination set you had, and as a result 1 is a type of solution that you can arrive at. Likewise, you have different solutions. 3 is another solution. So for reaching 3, the bacteria would take go straight down, and then it would take a right, and then it goes straight down, so it arrives at the exit 3. And then you have a special case of exit called as exit 4. And the exit 4 can be reached in 2 directions, 2 ways. Either the bacteria could take right, and at this split junction, it could take a left, reaching to 4. So which means it's adding 1, 2, 3, 4 elements. And in this here, the bacteria could take otherwise, first left, and then 3 lefts, and then continuously rights, meaning 1 plus 3 adding to 4. So this exit 4 can be reached by 2 different combinations, 0, 0, 4 or 1, 3, 0, which are basically the additional products. So by this approach, you would be able to solve complexity 2 type of subsystem problems. And likewise, the bacteria could explore the other solutions, namely solution 5, which is a combination of +4 and +1, or the solution 7, which is a combination of 4 plus 3. Or solution 8, which is a combination of 1 plus 3 plus 4 computing to 8. And the exit 2 and 6, they can never be reached at any point because there is no route that's possible. And if the bacteria is following the algorithm that is encoded in the split junctions and the pass junction and the joint junctions, they would end up in these outputs or in these exits, except exit number 6 and exit number 2. So using this approach, we were able to solve the subset sum problem up to a particular input numbers. So here we show one example of how bacteria would go through different numbers. So the first example is the split junction. The bacteria is taking the right. So this is a bacteria. So it's 0. And the next split junction, again, another 0. And it keeps going. The next split junction, which is corresponding to +4. And it ends up in the exit number 0. Likewise, the bacteria could explore other routes. And imagine you have a bacteria, that's one bacteria entering, and during the process of running in the network, it divides into 2. So you have one bacteria dividing into 2 and exploring 2 different routes. So imagine you have 1,000 solutions and you have 500 bacteria that's entering, and at some point each of the 500 bacteria divides. So what you have eventually is a doubling of the computing time, or sorry, doubling of the computing power. Okay, so if a bacteria is following this network, the junction rules like the split junction, pass junction, and joint junctions, then what you would have is bacteria ending up into the right solutions. And these right solutions can be exported or can be understood using the heat maps again. So this is a specific problem of 2, 5, 9, 3 elements. So 2 plus 5 plus 9 would correspond to the total exit of 16. And anything that red you see here is actually bacteria, actually the exits that cannot be reached at all. Okay, and anything that you see in green are the solutions that can be reached by following the rules in the junction. And you can very clearly see here from this specific density map that all the black regions corresponds to bacteria not exploring a particular route, and it is not possible to explore. And all the routes with different intensity of blue and different intensity of crimson or violet is corresponding to the right solutions. So if the bacteria is following the rules that you encode in the network, and if you have a bacterial candidate that could follow the rules, then what you have at the end is a perfectly operating bacterial based computers. This is another example. Instead of 3 elements, we here used 4 elements, 1, 3, 5, and 11. And it has a total of 20 exits, and 2, 7, 10, 13, and 18 are the wrong exits. In any way you add 1, 3, 5, and 11, you cannot come to a combination of 2, 7, 10, 13, and 18. And you have a much larger network. So this network is made of 5 elements, has a total number of exits of 42. And everything in red is a wrong exit. Everything in green is a right exit. If the bacteria is following the rules that you have incorporated, they are going to solve the combinations. And based on counting the number of bacteria that's coming out of each exit, you can plot a bar chart using which you can say if a bacteria has solved a problem or not. Here you see actually thousands of bacteria running in the network. So every white spot that you see here is actually a bacteria that's exploring the solution. And imagine this 16 solution here is explored by hundreds of bacteria. 20 solution explored by again hundreds of bacteria. 42 different solutions explored by again thousands of bacteria. So as the problem size increases, you solve with larger and larger number of bacteria because you have larger bacteria, higher number of bacteria entering the network. And at the same time, these bacteria that's entering the network, they could divide. And at the same time, increase the computing power as they solve the problem. Now the question is, when can we beat a computer by using such bacterial based computers or bacteria operated computers? So we did some simulations to explore how long do we have to wait in order for a computer to be overtaken. So what you see here in straight lines, diagonal lines, are the computing power of different ranges of computers, starting from 64-bit 1 gig 2.3 gigahertz computer to as slow as 32-bit 500 MB 660 megahertz computer. So this is something like 1990s computers, and this is something like 2018 computers. So these are the computing power, and other lines that you see here are the computing powers of different bacteria that we used, particularly E. coli and Vibrio nitrogens. So these two blue lines correspond to bacteria trying to solve without division. But the moment bacteria solves the problem with division, meaning they multiply in the microfluidic network that we are using, what happens is at around the cardinality or input element of number 35, we can see that they can easily overtake a computer. So this intersection line of the straight lines where the computing power of a computer is shown compared to that of the bacterial computer, that is what means the intersection point means these points a bacteria could overtake, or a bacterial operated computer could overtake the whole of the computers that we use in our everyday life. So we have advanced so much on this area in the last year that we have some evidence, strong evidence that even quantum computing could be easily overtaken by a bacterial computer. And the reason for all of that is because of parallel computation. You do a parallel computation, you have a technology or a device that could translate biology into a parallel computing method. Then what you have is a biologically operated parallel computing machine. So the whole point of explaining how a bacteria could solve a mathematical problem and could do parallel computing is, it doesn't mean that eventually computers would be replaced by bacteria. No, that's not the way. The idea is for some specific problems which are of very much of importance, particularly related to space exploration or trying to understand tractability of other bigger NP-complete problems, cryptography, random number generators, etc. You need to have a parallel system, and we are trying to establish that we could use bacteria for all those parallel computing approaches. So that brings me to the last part of my lecture, which is the conclusions. So the main take-home messages that I would like to give you is probably if you haven't been aware what is microfluidics, I am happy that I introduced you to what is microfluidics and how you do some fabrication. So everything that I talked about microfluidics, its fabrication, etc., is only less than 2% of what we give in an actual lecture that I handle. Then what I explored to you, what I walked you through is about bacterial motility and how we could translate motility of bacteria to understand its intelligence mechanisms, how bacteria negotiates with the geometry that's around it. And of course, I introduced you to something that's completely unique, I would say, which is how we could perform computations with bacteria. And for this, we specifically use a graph theory-based approach, which is a mathematical engineering-based designs, which is designing microfluidic devices and using bacteria to explore them and understand how the computation is being solved. So if you are interested on any of these aspects of research, you could look into the two articles which were published by our team, like one in 2016 and one in 2018. And you would be having a lot of research updates from our team in the following months through publications. And if you're interested in our labs, you can use the link I gave you there. You would know what active research that we're doing and any type of position that's available with us. So I would like to thank at the same time your managements, as well as specifically to our biological microfluidics team, headed by Professor Danby Nicolau, and our collaborators, close colleagues, Falco, Mona Lisa, Julia, Kabia, and other collaborators in Czech and different parts of the world. So those are funding agencies. So all of this biocomputation work is carried out in association with the DARPA, the Defense Associated Research Grant System that's operating in the US, and as well as with NSERC, and one of the recent grants that we have, which is New Frontiers for New Frontiers and Research Funds, which is an exploratory grant that we received for carrying out biocomputation related approaches. So that brings me to the conclusion of this slide. And one personal note that I would like to finish off is you could also follow the active research updates as well as important informations that I try to talk along with one of my friend Nirmal, who is also a PhD graduate doctor in neurosciences. At neurosciences working in London in the UK. So we run a channel called as Mayawa 423. It's a channel where we try to disseminate information related to science, biology, technology, etc. in Tamil. So you could visit this channel, you could subscribe it, you could have a look into the videos that we have posted. So get some benefits out of it as well. So again, I'd like to thank the St. Joseph's College of Engineering, the management and the organizers, Dr. Chamandishwari, and the students who have made this session possible. And thank you very much for bearing with me for the past 1.5 hours. Thank you very much.
Thank you very much, sir. It was a very informative session. I'm pretty sure many of us learned about microfluidics and bacterial motility, and obviously bacterial computers. And we were exposed to new concepts that we never knew before. We actually have two questions here, so from YouTube. So I'll ask them to you, and then you can answer them. Is that fine?
Yeah, sure, go on.
Okay, so the first question is, what type of softwares are used for identifying bacterial intelligence?
Okay, so that's more about image processing that we use. So most of the time we use freeware. Most of the time we use softwares that are available, open for the public, used by mostly the research community. One simple software that we use is called as ImageJ or Fiji. So this is an extensive platform for image post-processing of images that you get from microscopes. And most of the time, we use several plugins that are associated with this openware. For example, for tracking, we use plugins like mTrackJ or TrackMate, etc. And there are also other commercial softwares like Imagina, etc., which you can use if you are willing to pay money and buy the softwares. But I think in terms of science and the research community for image processing, etc., the community is quite open, and you have several different plugins that you can insert into ImageJ to make your need. Also, we have special computer scientists that are computer science students that we are associated with at the McGill University, who try to create separate plugins or separate research tools that would fit our needs for understanding the bacterial motility patterns. So the whole concept is, you understand the bacterial motility, probably you will get to know about how the intelligence in them functions. So in that angle, if you look, most of the time the softwares that we use are trying to understand what velocity bacteria goes, where it goes, how quick it goes, how fast it goes, how quick it goes, or how slow it goes, etc. So for all that, we use ImageJ and the related plugins. Probably I can provide you the links where you could download the software and you could probably experiment it with a bit.
Okay, thank you, sir. The last question is, does the bacteria of the same species have the same type of intelligence?
So in a way, so far with what the 5 types of microbes that we used here, we were not able to confirm that. Because I showed you an example with Vibrio nitrigens and Vibrio fischeri. This is a Vibrio type of bacteria. It belongs to the same genus but different species, and the way they behaved were completely different. For example, Vibrio fischeri was trying to stay in the center of the microfluidic devices, while Vibrio nitrigen tried to go towards the edges of the device, towards the walls of the device, or wall following and wall repelling. So from my understanding, based on the 5 species that we have used, it's very difficult to say. Probably there would be, but we for sure know that there are different genus of bacteria which could behave the same way. For example, E. coli and Vibrio nitrigens behaves the same way. They both of them try to follow the walls. Sorry, when it tries to go through the networks. So what I would say as an answer for this question is, between the genus, we haven't explored any that is very similar. But between the species, we haven't explored anything that's similar. But between the genus, yes, we have seen bacteria with similar type of exploring nature. So in that way, you can call their intelligence might match to some extent as well.
Thank you, sir. We actually have one more question now.
Yeah.
Okay, so the question, I'm sorry, just give me 1 second.
Yeah, sure.
Okay. Yeah, so the question is, does 3D printers help in furthering this technology?
Yes, so that again depends on the type of resolution that you want to achieve. We have been exploring 3D printing for making such devices, but if you actually look into 3D printing, the best of technology now could give you a resolution anywhere to 2, 3, maybe 4 microns. If you want to go below 4 microns, it would be very difficult. But for all of our microfluidic devices that I'm using a resolution close to hundreds of nanometers. Because if the surface of the microfluidics is very rough, the bacteria will not move smoothly. It will try to stick to the surface. So I need to have the surface as smooth as possible. So because of this specific reason, I'm using e-beam based lithography, while even 3D methods could be used. But if the resolution is not an important parameter. And we have one of the project that we are currently running is trying to explore the use of Euglena in a 3D biocomputation device. So yes, 3D printers can be used, and because Euglena cells are quite larger, like 10 to 20 microns in size, you can easily use a 3D printer for that.
Thank you, sir. We actually have one more question, and with this, we'll wind up the Q&A session.
Yeah.
What happens if the bacteria doubles in their multiplication while solving the maze?
The time, okay, that is dependent on how much time you use the computation device to operate. When it comes to maze, typically we would solve the small maze that I showed you in few minutes time, like let's say 3 minutes to 4 minutes, you will see that bacteria finding the solutions very quickly. But when it comes to subsets and problems, if the network is very large, definitely multiplication would help. And the same ideology would apply to mazes as well. Imagine you have a very large maze and the bacteria at least find one solution it needs to explore 1 hour. And in this scenario, if the bacteria could multiply, that's good. You can let the bacteria multiply and explore more and more of solutions, and that's very good as well. So yes, even if the bacteria multiplies in the maze, you could have a higher rate at which you could find the solutions. So that will be a good thing as well.
Thank you, sir. Actually, we have some more questions here. So do you have the time to answer some more?
Yes, go on. Yeah, no problem.
Yeah, sure, sir. Thank you very much, sir. The next question is, can we incorporate certain corrective chemicals, per se antibodies, into the notion of microfluids?
I'm sorry, can you please repeat the question? I'm sorry, I didn't listen to it properly.
Yeah, sure, sir. Can we incorporate certain corrective chemicals, antibodies, into the notion of microfluids?
Yes. So so there are, you can definitely do that in microfluidics. If you want to introduce a specific chemical signal, or if you want to introduce a specific let's say an antibiotic, or let's say attractant like glucose molecules, etc., at specific points in the network, yes, you can do that very easily. What you do is you have specific nozzles or holes punctured onto the device wherever you need, and through that you could inject or you could dispense the solutions very easily. So that's doable as well. We are trying to explore this technology or this technique because if the microfluidic network is large, and if the points where you need to dispense the solution is, let's say, 100 microns or 200 microns or 50 microns in size, you can use micro needles to do that. But if the points where you need to inject is much smaller, like let's say 1 micron or 2 micron or 3 microns, in this situation, you need to fabricate another layer of device that would fit onto the first layer of device. So in this situation, it would be a bit difficult, but it is doable. And there are already a lot of people who use similar such techniques for droplet microfluidics, where they try to generate droplets by letting fluid flow in different directions. So it is doable, yes. But we haven't tried it. At least in our lab, we haven't tried it. Probably some other lab in the world has expertise in doing that.
Okay, sir. Thank you, sir. The next question is, can you elaborate on the applications of wall-dependent and wall-escaping bacteria? Okay, so
this is something that you can try to how could I put it? Okay, so imagine a bacteria which is causing, okay, so I'm trying to go out of a context with respect to biocomputation, because in terms of biocomputation, we like to use wall-dependent bacteria because they try to avoid U-turns. Imagine a bacteria is trying to solve, and towards the end it makes a U-turn and comes back to the exit, to the input point itself. So then that's useless of using the bacteria. So in terms of biocomputation, there are applications with respect to wall-dependent movement of the bacteria. In terms of wall-escaping bacteria, we have another project that also I'm running, which I'm running along with my team, which is basically simulating maps or simulating traffics using bacteria. So that could also be for that situation. We don't need a wall-dependent bacteria. We can use any bacteria that tries to escape from the wall as well. So that will help there. This is in terms of biocomputation or biosimulation related applications. But if I have to look a bit outside the box here, or what otherwise people could use for diagnostics, etc. If you know how a bacteria is moving, like let's say wall dependent or wall escaping, you could we have some simple geometries which I haven't shown in my presentation due to time constraints, where you could efficiently trap even the wall escaping bacteria. So such type of devices, microfluidic devices, could be used for trapping bacteria, or to some extent even to trap circulating tumor cells, etc. So the circulating tumor cells in a microfluidic device, they do not move by itself, but in the fluid flow they can move, and they would possess all the properties that the fluid has, or like a Newtonian and non-Newtonian fluids. So what I think here is, when it comes to answering to the point, in biocomputation, biosimulations, yes, wall dependent and wall escaping bacteria has a great influence. We like to use wall dependent bacteria for biocomputation. And in terms of general applications, if you know the property how the bacteria would behave towards the walls, then technically you can prepare devices that would enhance it to trap the bacteria that you can use later for diagnostics or even for single cell studies, etc. So yes, depending on what type of bacteria you use and what type of microfluidic network, how they behave to the walls, you can use it in different ways.
Okay, sir. Thank you, sir. With that, we'll have to conclude the Q&A session due to time constraints.
Yeah.
Yes, sir. Thank you once again for taking the time to share your knowledge and research, and also thank you for taking the time to answer some of our participants' questions, sir. Thank you very much.
Sure. Yeah. Thank you very much.
Okay. Now I call upon Dr. Chamundeshwari to deliver the vote of thanks.
Thank you, Swatika. I hope everyone has enjoyed this informative and unique lecture about microfluidics, bacterial intelligence, and biocomputers. Wow, we have a wonderful bacterial world and bacterial computers. I would like Dr. AYYAPPASAMY SUDALAIYADUM PERUMAL, who is a postdoctoral fellow researcher engineering from MICROFLUIDICS University, Canada, for spending his valuable time to join us for this session, BioTalk Series Week 5, and for delivering his important work and research aspects today. My personal thanks to AYYAPPASAMY SUDALAIYADUM PERUMAL once again. I would like to take this time to thank our patrons of St. Joseph Group of Institution, Dr. B. Babu Manoharan, Chair, Jessie Priya, Managing Director, Mr. B. Shashishekar, Director, and Dr. Vaddi Shashagiri Rao, Principal, and Dr. B. Team Research for giving us the continuous support. I would also like to thank our beloved HOD, Department of Biotechnology, Dr. G. Sri Kumar, and our advisor, Dr. Renika Vishwanathan, for provoking us to implement this Biotech Series talk and providing their continuous guidance to make this event to happen. I would happily like to appreciate and thank our final year student organizers, Ms. Niveta, Ms. Swatika, Ms. Satvika, Ms. Madhubala, and Mr. Kumaran and Mr. Raghav for organizing the whole event and making it successful. And finally, I thank all the participants for your kind cooperation and ensuring an active participation to make this event successful. Once again, I thank all the participants for kind listening. Thank you all. I hand over the session to Swatika.
Thank you, ma'am. Before we wind up for this session, I'd like to remind you all that the feedback link has been posted in the chat box, and it is necessary to fill it up for receiving the e-certificate. Ensure that you enter the correct details, as they will be used to generate your certificates. Once again, I'd like to thank you all for attending this session. I hope to see you all in future events organized by the Department of Biotechnology from St. Joseph's College of Engineering. Until then, take care and stay safe. Thank you.
Thanks.